In this video, we will discuss the final figure about this paper or the final experiments that they have presented in order to prove their point that DiaBZl will help in the tumor suppressions or tumor regressions. So what they did in this final experiment is that they did their DiaBZl treatment in the mouse model that is infected with this MEL cancer cells. And in the previous video we have understood that in the in vitro experiment they have seen that DiaBZl will cause this sting agonist on the tumor cells that will help the engineered T cell receptors to recognize the tumor cells and trigger apoptosis in those tumor cells. So after validating their hypothesis in vitro, they wanted to check whether this hypothesis also works in vivo or not. So they took the mice model, NCG mice models, and infected them with the tumor cells, and then used these DiaBZls for the increase in the antigen presentations on the tumor cells that can be detected by the immune cells. So in the panel A, what they have shown is that they have shown the the difference between the mice models, uh, in which in one case where mice models is treated with the DiaBZl, in other case mice models is not treated with the DiaBZl. Sorry. So in the mock case, they can see that low amount of this NY-ESO antigens and this HLA. HLA is the part of major histocompatibility complex that, or we can say it is part of the immune system that helps in the recognition of the antigens that are presented on the tumor cells. So in the monks we see low amount of this HLA, the low amount of this antigen presentations. However, after the DiaBZl treatment, we can see that there is a increase in the amount of both the HLA and also the antigens, that is NY-ESO-1 antigens, and these things are quantified in the panel B, where we can see that after DiaBZl treatment there is a significant increase in the NY-ESO-1 signals, and also there is significant increase in the HLA signals. And these things and after validating this immunohist after validating that there is increase in the antigens because of this DiaBZl's treatment through the immunofluorescence, they check whether there is a change in the apoptosis in both the samples or not. And what they find out in the case of mock, they see almost negligible amount of apoptosis. So they measured apoptosis using the proteins that is cleaved caspase 3. So they check the cleaved caspase 3 signals in the mock, and they do not find any signal, or very negligible signals are there. Whereas in the case of the DiaBZl treated mouse, they see a significant increase in the numbers of the signals of this cleaved caspase 3, and these are shown here like in the dark brown color. And the quantifications of this panel C is in the panel D, where we can see that the DiaBZl treated mouse models there is a significant increase in the cleaved caspase 3% compared to the mock case, indicating that this DiaBZl helps in the tumor apoptosis. And thereafter, after validating that there is increase in the apoptosis in the DiaBZl treated mouse models, they wanted to check the reasons why there is a increase in the apoptosis. So when they check the literature, they are able to find that this p-STAT1 helps in the antigen presentations to the immune cells. So they hypothesize that these DiaBZls might cause increase in this phosphorylation of this p-STAT1. So they check the p-STAT1 in those samples, and what they find out is that in the case where T cell receptors are treated with the DiaBZls, and they see that increase in the p-STAT1, and when they use the DiaBZls only, they also see the increase in the phosphorylated p-STAT1. And when they use the combined conditions, that is T cell receptors that are treated with the DiaBZls and the DiaBZl treatment, they see that there is a increase in the phosphorylated STAT1. And however, when they use in the non-control, in the control case, and also in the T cell receptors In the control case, they see the very minimum amount of phosphorylated p-STAT1, whereas in the T cell receptors they see slightly increase, but those increase is not enough compared to the other cases. So indicating that, and so this results is also in line with the previous literatures that phosphorylated p-STAT1, increase in the phosphorylated p-STAT1 will cause apoptosis in the tumor cells, as they have found the apoptosis in the DiaBZl treated cells and the increase in the phosphorylated p-STAT1 in those cases. And the quantifications of this panel E is in the panel F, where we can see that in the case of these DiaBZl treated T cell engineered T cell receptors and the DiaBZl's injections into the mice models, they see that a maximum amount of phosphorylated p-STAT1, or when only a DiaBZl treated samples are used, these two seems to be comparables. So from these experiments, these final experiments, they conclude that their DiaBZls will cause or will improve the antigen presentations in vivo models also, in vivo mouse models also, and then cause that will results in this apoptosis, increased apoptosis of the tumor cells by the immune system. So I hope you understood how they have made a point that this sting agonist DiaBZls will cause apoptosis in the tumor cells. If you need to understand more about their experiments in detail, you can go through how they have quantified this, immunofluorescence image using ImageJ. You can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.